It's about 9, I think, in the morning.
Yeah, 9:30.
Now in Indonesia, it's 11:30. So it's
9:30, 9:30.
okay. So I hope also this program can be powerful tools for the student to self-development and also for planning the future and maybe planning the research based on this lecture. So please, I will ask Prof. Uun first. Prof. Uun. No, Prof. Uun is not here. Prof. Uun.
Hello. Yes, thank you, Miss Lutfi. Good morning, Prof. Prakash, and yes, for the join and enjoy for the join with us with the 3-in-1 program. So I'm sorry. I am not together with you for so long to the 3-in-1 programs before today. So maybe for the next I will do the join with the program today and after that until for the 28th September. Maybe weekends our study program can mix the collaboration with you and your study program in India maybe. So after that, I hope you can come and arrive to Indonesia. Thank you, Mbak Lutfi. Monggo silahkan. Thank you, Prof. Prakash. Okay.
Thank you so much. So,
Okay, please start the lecture. Dr. Prof. Prakash will give the lecture about the gene expression and regulation.
yeah, thank you, madam. Thank you. Thank you, Lutfi, and thank you, Professor Won. I think is it audible to everyone? Also, can you see my slide? Is it okay?
Yes, of course.
I think the slide is moving now. Is it okay?
We can see clearly the slides.
Okay, madam. Thank you. First of all, I would like to thank Professor Won as well as Professor Lutfi for giving this wonderful opportunity for me. So today my topic is gene expression and regulation. I should thank both of you as well as the university for giving this wonderful opportunity once again. So I'm really sorry, as well as I would like to sorry to all for delaying for this late session. I think 30 minutes I'm late, so sorry. So okay, today I'm going to talk about the gene expression and its regulation because as a biotechnologist, as a molecular biologist, this gene expression studies is one of the important part to understand in molecular level mechanism as well as if we need to change anything in this genomic level. Already, I'm talking about the genome mapping in the last session. I hope if you are able to participate in the last session, you may understand what is meant by genome mapping, as well as it's a continuation process. Actually, okay. So first, let us see about what all the content I'm going to today. I'm going to talk about this gene regulation and its gene expression and all. This transcription and translation and regulation of the gene expression and lac-operon theory. So these are the things we are going to discuss today. So first, let us see about the gene expression. So before I'm going to talk about this gene expression, I just want to ask you one thing. Already I may discuss in this topic, but even though once again I'll just ask you, just you think about yourself. You don't need to answer me anything. So just think about yourself. In 26 letters of in English, I think you all know very well that English letters. There are 26 letters of there. Do you know how many stories we can able to write by using these 26 letters? We cannot say this many numbers only. We can able to write the story because each and every stories are they are having the unique things, unique theme maybe there. But the actual thing is all the different type of stories, even it's maybe a crime story or love story or adventure, whatever it is. But we can able to write only using the same 26 letters. But the only difference is the arrangements of the words, arrangements of the sentence, arrangements of the order. So that's only classify the nature of the nature of the story. Likewise, here as a molecular biologist, we must know this adenine, guanine, cytosine, and thymidine. All these four nucleotides is going to uh, create a new species based on the arrangements of these amino acids or nucleotides. Based on these arrangements, it's maybe any n number of genes they can able to produce, as well as the n number of species they can able to produce. It's all depends upon arrangements of the nucleotides as well as the size of the nucleotide base pairs, number of base pairs, and as well as arrangements of the nucleotides. This is responsible for each and every chromosomal species diversity. So now let us see about this gene expression. Is it anything audible, or if you have any problem with my slides or anything, you just text me, or else I can go ahead. Okay, so the gene expression is the process by which the information encoded in gene is used to direct the assembly of the protein molecules. So the synthesis of the protein from the nucleotides, which means the genes. Before that, we must know each and every species. It's contain the chromosomal DNA as well as the plasmid DNA or extra chromosomal DNA. So otherwise, simply we used to call as extra chromosomal DNA. Sometimes we used to call as plasmid DNA or mitochondrial DNA or something like this. But each and every chromosomal DNA or plasmid DNA, whatever it is, it's contained thousands of genes. The number of genes, it depends upon the species size. In case of prokaryotic size, the number of genes may be very less in number. Likewise, in case of eukaryotic cells, it's contain the large size of the chromosomal DNA. So it's contain the thousands, more than 1,000 or 5,000 number of genes. It depends upon the size, chromosomal size as well as the nature of the species. So, we must know how to express all the proteins simultaneously or step by step. Okay, it's explored through the study protein structure and functions, transcription through the transcription and translation. So all this process, gene expression, is based on transcription as well as the translation. Is the process by which information from gene is used to synthesis of functional gene product. So let us see how it's work. So genomic DNA is contained double-stranded DNA. It's both contain the promoter region, operator region, and terminator regions. Everything it should contain, as well as last region itself. I told you each triplet codons are there. Each triplet codons are code for one amino acids. That one amino acids are controlled by the codons and anticodons and all. There are so many processes are there. Now let us see one by one. So here the genomic DNA is contain exons and introns. So exons and introns it's alternative gene sequences which is present in this genomic DNA. So from the genomic DNA, we can able to synthesize the RNA through the transcription. Through the transcription process, we can able to synthesize the RNA. So by this way, after synthesis of this transcription or mRNA or RNA, tRNA, whatever it is, the post-transcriptional modification will take place after this process. So first, the initial process, the conversion of the genomic DNA to RNA. Before that, already one more process that is there. It's called as replication. Replication, I hope you all know already, that synthesis of DNA to DNA. The double-stranded DNA is converted to single-stranded DNA. Again, it's going to synthesize a new strand. So both strands are become the two daughter strands. So again, it's going to multiply based on the replication process. It's the process of replication. So likewise, the transcription process also takes place after when the gene expression starts. So after the transcription, post-translational transcriptional modification will take place. Then it's a splicing process. It's a mature RNA. Only the introns are joined together. Exons are removed from this introns. So after splicing, the mature mRNA is going to synthesize the protein. The proteins also after synthesis of the protein, the post-transcriptional relational modification also takes place after synthesis of the protein molecules. Okay, this is the entire mechanism of gene expression studies. So now let us see one by one. Genetic information are chemically determined by DNA structure is transferred to daughter cells by DNA replications. This was already I told you, and expressed by transcription followed by translation. This series of events are called the central dogma theory. Central dogma theory, I hope you all know very well. The DNA to RNA, RNA to protein, DNA to RNA, RNA to DNA. This process is called as central dogma theory. Is found in all cells and processed proceeds in similar way except retrovirus, which possess enzymes reverse transcriptase, which converts RNA into complementary DNA. For an example, they will tell you retrovirus, but not only retrovirus, most of the RNA viruses, they don't have the DNA. So they should convert the RNA to double-stranded cDNA. Then they can start the process of replication, transcription, and translation process. So here the simple process in the imaginary view, you can see transcription as well as the translation process. So this is the extended central dogma molecular biology, includes all the cellular process involved in this flow of genetic information. So this gene expression, it's contained only the two steps. First one is the transcription and second one is the translation. Already we discussed about something about this transcription as well as the translation. It's together, they are known as gene expression. During this process of transcription, the information stored in a gene's DNA is passed into similar molecules called as RNA in the cell nucleus. Just an example I'll give you. For an example, if you want to take a photocopy of your textbook, because textbook is it's act as a parent. So from the textbook, we are going to take a copy of the textbook or whatever we need, even the particular pages if we need, we are just going to make a copy from the textbook. Likewise, the DNA, or synthesize the RNA. That RNA is carrying the information from the DNA. This carrying the information from the DNA, chromosomal DNA. So this is the way they are transferring the information from DNA to protein through the way of mRNA synthesis. So now let us see the transcription process. So transcription takes place in the molecules nucleus. It uses the DNA as a template to make the RNA. This is called as mRNA molecules. During the transcription, a strands of mRNA is made that is complementary to strands of DNA. This transcription takes place in the three steps: initiation, elongation, and termination. So already I told you the transcription process means the synthesis of mRNA from the DNA. DNA is act as a template. So from the template, by using the template, they can able to produce the messenger RNA is carrying the message from the DNA with the help of the triplet codons. Already we discussed about this each and every codons for the protein synthesis. Likewise, each and every the mRNA is just a complementary strands for the DNA that except thymidine. We can synthesis we can make use of the uracil by the replacement of the thymidine. Or by the synthesis of the mRNA, they are not going to utilize the thymidine. Just because of the thymidine, here we are using the uracil for the mRNA synthesis. So the first step one, is initiation process. So initiation is the beginning of the transcription. It occurs when the enzymes of the RNA polymerase binds to the region of gene called as promoter region. So the RNA polymerase should go and bind in this promoter region. This signal the DNA to unwind it. So the enzymes can read the base in one of the DNA strands. The enzymes now ready to make a strands of mRNA, which are complementary sequence of bases. For example, once again, I tell you that RNA polymerase, it helps to initiate the process of transcription. So this RNA polymerase enzymes go and bind in this promoter region of the DNA. That means the gene. So after this binding of this RNA polymerase in this promoter region of the DNA, it starts unwinding the double helical strands. So the RNA polymerase can able to read the bases. So then only they can able to read. As a double-stranded DNA, the RNA polymerase cannot able to read the sequences. So this is the flow, or this is the imaginary explanation for this process. So initiation process starts in the promoter region. So the RNA polymerase go and bind with this in this promoter region. Then they start the unwinding of the double helical strands. So it's a wobble hypothesis. Simply, it's to create like an I structure. So they can start the process of uh, transcription. So here, the RNA synthesis takes place with the help of RNA polymerase. So the next one is the elongation process. Step 2 is the process of elongation. Is the addition of nucleotides to the mRNA strands. The RNA polymerase read the unwound DNA strands and builds mRNA molecules using the complementary base pairs. There is a brief time during this process when the newly formed RNA is bound to the unwound DNA. During this process, adenine in the DNA binds to an uracil in the RNA. Already told you, adenine cannot able. We are not here. The mRNA may not use the thymidine. By the replacement of the thymidine, they are utilizing the uracil for mRNA synthesis already because we know the difference between the RNA as well as the DNA is an uracil as well as thymidine. This is another major difference between RNA and DNA. So this is the process of elongation process. The next one is the termination process. Is the ending of the transcription and occurs when RNA polymerase processes the stop codon, stop sequences. So stop sequences in the gene, the mRNA strands is completed and detached from the DNA. Is the termination process. So once they reach the stop sequence, that means the stop codon or the stopping gene sequences. So once they reach, this entire process will be terminated by this process, so by this RNA polymerase. This is the way they are stop the process. So likewise, the translation process. So once the transcription is completed, then I already told you the protein post-transcriptional modifications also there. I'll explain in detail about this post-translational modification later. So now let us see the translation process. This is the process by which the proteins are synthesized. The translation is a complete cellular process with mRNA molecules, ribosomes, tRNA molecules, amino acids, energy source of ATP and GTP, and number of factors act together in highly coordinated way. This translation process is a very big process. There are so many factors and elements are required or involved in this process of translation. One among the what I already told you, the ribosomes, tRNA molecules, amino acids, energy sources, ATP and GTP, a number of factors, the initiation factor, elongation factor, something like this. So there are so many factors also responsible for this process. So this translation is the process in which ribosomes in the cytoplasm or the endoplasmic reticulum synthesize the proteins after the process of transcription of DNA to RNA in the cell's nucleus. So the transcription process. So before understanding this transcription, translation, and replication, we must know where it takes place. The replication always takes place inside the nucleus in case of eukaryotic cells. But in case of prokaryotic cells, everything happens only in this nucleoplasm. But in the cytoplasm. But in case of the eukaryotic cells, there are two different regions are there. One is nucleoplasm, another one is cytoplasm. So here, an example, the DNA was located in this nucleus. So the mRNA synthesis should be takes place only. They can able to synthesize the mRNA only inside the nucleus or nucleoplasm, inside the nucleoplasm. But in case of mRNA, this protein synthesis, so after synthesis of the mRNA, sometimes it may takes place in this endoplasmic reticulum or else cytoplasm. So there are two regions. There may be the transportation of the mRNA from nucleoplasm to cytoplasm that will take place inside the cell. So we must understand what are the different regions are there, what are the process that takes place, and where it was takes place. Either the entire process will take place inside the nucleus, inside the nucleoplasm or cytoplasm. So here, likewise, the different stages are there in this protein synthesis also. First one is the initiation and elongation and termination. So initiation process already we know is ribosomal subunits bind to mRNA. Then after the binding of this mRNA in this initiation site, then the elongation process takes place. The ribosomes move along with the mRNA molecules, linking the amino acid because each and every ribosomal unit it's contain one amino acids. and all these amino acids are formed together. Then they join together with one by one. They form a polypeptide chains. So after this synthesis of this polypeptide amino acid chain, the termination process will take place once they reach the stop codons. Already told you there is a stop codon, there is an initiation codon. So initiation codon helps to initiate the process, the starting point of origin of origin of translation. So likewise, the stop codon, is the termination codon. This helps to stop the process of the protein synthesis. This is three simple steps. Actually, I'm going to elaborate in detail the same process. I'll show you the some videos also, so that you can able to easy to understand. So this is the three major steps involved in this translation also. So this is the process in imaginary view. So first, there are two subunits are there in this ribosomes, smaller subunit and larger subunits. It may be size may be varied, 30 subunits or 50 subunits. So, but they should classify into two types: only smaller subunit and larger subunit. So first, the smaller subunit, the mRNA should go and bind in this smaller subunits. Then the larger subunits are come and bind in this smaller subunit of ribosomes, ribosomal rRNA. Then the tRNA is go and bind in this specific anticodons, because the tRNAs contain three triplet codons. That mean AUG, ATG, AAG. There are different type of codons are there. Already we have seen in the table format. So each and every tRNA it's contain codon regions as well as the similar anticodons are will be present in this mRNA sequences. So based on the mRNA sequences, they choose the codons. They choose the tRNA. So based on the tRNA, each tRNA contains one amino acid in the top end. Also here there are two sides of the P sites and S side. So based on this attachment of these smaller subunits and larger subunits on the tRNA, the entire complex will be formed. Then they start the moving towards opposite directions. So the tRNA is going to move the next position. So again the next tRNA, that means another tRNA, they can able to bring one amino acids, which is which may be methionine or erythrocyte or whatever it is, among these 20 amino acids. Each and every tRNA may bring one amino acid, as well as they'll go and bind in this specific location of specific anticodon on which is present in this mRNA sequences. So this bonding will take place in this ribosomal entire translational unit. So likewise, it's moving one by one. So this process is called as elongation process. So once the process will take place, each and every amino acids present in this tRNA top end, so they can join together, which each the top layer. So they can form, yeah, they can able to synthesize the new protein sequences um, based on the tRNA as well as the mRNA, which is contain the codon and anticodon regions. So based on this codon, they can able to form amino acid chains, so they can synthesize the entire protein synthesis. So before going for this regulation, I just want to show you one video. Just wait. Just a few seconds. I just want to show you one video.
to produce a polypeptide. Some bonds come together to form 50 to 250 post. In transcription, the DNA within a gene codes for an mRNA, which then undergoes post-transcriptional modification. Each end gets a special cap tacked on, like the 5' cap, which is a modified guanine, and the poly A tail, which is 50 to 250 adenines. Then a protein complex called a spliceosome will cut out sections called introns, and other sections called exons come together to form a smaller mRNA, which then moves from the nucleus to the cytoplasm. This is where translation occurs. In translation, the mRNA, a ribosome, and many tRNAs work together to produce a polypeptide. Some of these polypeptides will then be complete, but others will instead undergo folding, likely in the endoplasmic reticulum, and sometimes modification in the Golgi apparatus, where groups like sugars, lipids, or phosphates are attached. Then the proteins are delivered to where they need to go. This sum process, otherwise known as the central dogma of molecular biology, illustrates how gene expression generates all of the proteins in your body. Once again, please watch my biochemistry tutorials on one expression. We know from learning about mitosis that every cell in your body, except your gametes, contains all of your genetic information, and therefore all of your genes. But different cells in your body serve different purposes. Some are muscle cells, some are nerve cells, some are liver cells. So different cells need to express different genes. How does a cell know which genes to express and which to leave dormant so that it can serve its particular purpose? This is done through regulatory mechanisms. These evolved very early in the timeline of life on Earth because single-celled organisms had an advantage if they only expressed the genes that code for proteins that are needed by the cell in a given moment. If a particular resource that the organism needs is plentiful nearby, it should stop self-producing that substance to save energy. If it is sparse in the environment, it needs to kickstart production to survive. This kind of metabolic control is self-regulating because the products of certain enzymatic pathways act as inhibitors for those pathways. So if there is a lot of a certain metabolite accumulating in a cell, it slows down the pathway by which it is generated. This is called feedback inhibition. But how exactly does this work on the molecular level? Well,
don't skip the opportunity to study abroad. With the Duolingo English test, we can make this process a lot less.
Well, bacterial cells utilize operons, so even though eukaryotes don't have these, they will be important for us to understand. Operons work like this. Let's look at a particular metabolic pathway present in the bacterial species E. coli. The amino acid tryptophan is synthesized in 3 steps, with each step catalyzed by a different enzyme, and it takes a total of 5 genes to produce these enzymes. These genes are found very close to one another on the bacterial chromosome, and a single promoter serves them all, producing one huge mRNA that produces all 5 enzymes during translation, when ribosomes anneal at any of the various start codons on the chain. This means that these 5 genes are coordinately controlled. Any influence on the transcription of these genes will impact the production of all of these enzymes. There is a segment of DNA, in this case in between the promoter and the first gene, which operates as an on-off switch. This is called an operator, and it controls whether RNA polymerase has access to transcribe or not. The promoter, the operator, and all these genes are altogether called an operon. Normally, the operon is on, but something called a repressor can bind to the operator, which then blocks access to the promoter, so RNA polymerase can't do its job. If the genes can't be transcribed, the enzymes can't be produced, and the cell can't build tryptophan. This repressor is specific to this operator, so it doesn't do anything to other genes, and it is a protein, which is a product of a different gene somewhere else in the DNA. This tryptophan-specific repressor is produced regularly, but in an inactive state that has little affinity for the operator. When tryptophan binds to the active site of the repressor, it changes shape to become an active form that has much more affinity for the operator, so it will bind and stay on for quite some time, thus turning the operon off, inhibiting gene expression and limiting further tryptophan production. The more tryptophan there is in the cell, the more repressors that will be activated to inhibit gene expression. The less tryptophan there is, the less inhibition there will be. While we just saw an example where a gene is typically on unless repressed, there are also genes that are typically off or silenced unless activated. In E. coli, again, there are genes that, when expressed, produce an enzyme that will metabolize lactose, a disaccharide, into individual monosaccharide units, glucose and galactose. There is typically a repressor bound to the operator that corresponds to these genes, but an isomer of lactose called allolactose will bind to the repressor and deactivate it, thus allowing for transcription of the gene, enzyme production, and higher levels of lactose metabolism. These two examples both demonstrate negative gene regulation. One repressed gene expression and the other deactivated a repressor, so the signaling molecules do not interact directly with DNA. There can also be positive gene regulation, where a signaling molecule like cAMP will bind to a protein called an activator, which will then bind to DNA and directly stimulate gene expression by increasing the affinity that RNA polymerase has for the promoter. So negative and positive gene regulation are both methods by which signaling molecules interact with operators, repressors, and promoters to regulate the frequency with which certain genes are expressed. Regulation gets more complicated than this, however. Many cells need to do more than respond to levels of glucose or lactose. When a fetus grows, cells are dividing and becoming specialized, and each cell acquires a distinct role on the basis of selective gene expression. Nerve cells and liver cells and skin cells are very different from one another, even though they all possess the same genetic material, and the secret behind this is strict regulation of gene expression. In any given cell, some genes are expressed and some aren't. An easy way to turn genes on and off has to do with the way that DNA is wrapped around histones to form nucleosomes. When bound to histones, genes can't be expressed. In order to express a gene, the gene must become accessible. This can happen if an enzyme modifies a histone through acetylation, methylation, or phosphorylation, thus decreasing its affinity for DNA. When a gene is no longer coordinated to the histone, it is available for transcription. In order for transcription to proceed, proteins called transcription factors are necessary. Some of these bind to a section of a promoter, usually in a region called a Tata box, as thymine-adenine pairs are easier to pry apart, given that they make one fewer hydrogen bonds than a CG pair. Binding to DNA occurs due to a binding domain that has affinity for a specific sequence of nucleotides in the promoter. The transcription factor also has an activation domain, which will bind to other regulatory proteins that enhance transcription. A transcription factor can have one or more of either of these types of domains. In addition, there are other control elements farther away from the gene called enhancers that interact with proteins called activators. When activators bind to the enhancer, another protein can bend DNA to bring the activators closer to the promoter where the transcription factor can be found. Other proteins mediate interactions that produce the complete transcription initiation complex, which allows RNA polymerase to do its job. So we can see that transcription is quite a bit more complex than we previously discussed in biochemistry. There are many proteins involved when any gene is being transcribed, and so regulation of the levels of these proteins can regulate the expression of other genes. Some genes can only be transcribed when specific activator proteins are present, and this may only occur at a specific time, like hormones carrying a message to promote the expression of genes whose products trigger development during puberty. Combined with the acetylation and deacetylation of histones to either activate or silence genes, proteins that bind to mRNA to prevent translation, and other phenomena, the cell has several strategies at its disposal to regulate gene expression. A combination of these regulatory strategies therefore allows a relatively small number of inputs to regulate thousands of genes independently. Although these interactions are much more complex than we have depicted here, they tend to follow these principles, and with a basic understanding of both gene expression and cell division, we are now ready to look at more complex systems. Let's move on to some of these now. Thanks for watching, guys. Subscribe to my channel for more tutorials. Support me on Patreon so I can keep making content. And as always, feel free to email me, ProfessorDaveExplains@gmail.com.
Okay, guys, once again.
A virus. Contrary to popular belief, viruses are not alive. They are much smaller and simpler than single-celled organisms like bacteria, and they do not meet most of the criteria that biologists agree are required to call something alive.
Sorry. Okay. So I hope you can able to understand this gene regulation of the gene mechanisms, as well as now I'll tell you some extra points to you. So some of the environmental and cellular influence also regulate the gene mechanism, gene expression regulation. So once again, I tell you the gene regulation mechanisms. So of course, one of the complex process that determine when and how much gene is expressed and how they are the key points you can able to cover in your presentation cover. So gene expression always requires the regulation, or else what will happen? Because most of the cancers are is responsible for this kind of gene regulation mechanisms. So our absence or regular regulation mechanisms. So even the apoptosis, that means the programmed cell death, that's also just because of this the absence of gene regulation mechanisms. Okay, so now I'll just give you some small brief about the gene regulation once again, because the video point of view, I don't know, I hope you can able to clear, but even though I just want to give you small outline of this gene regulation once again. So first of all, the definition of the gene expression is the gene expression regulation refers to the mechanism that control the amount and timing of the gene expression. So when the genes should be expressed and how much of the protein required for this systems, that means the species. 1 second. So, for an example, I'll tell you, in case of any antigens enter in our body, for example, you can take an example of the COVID-19 or whatever it is. Any antibiotics, if it is any antigens come in, enter inside our body system, our immune system should produce the antibody against that particular antigens. So, first of all, they should analyze about the antigen, how many antigens are enter inside our body, and how much of the what are the type of antibodies are they we have to produce by the memory cells or immune cells and all. So how much quantity we require and when it should produce the antibody. So it's required some time to analyze the antigens. So based on the analysis report, they have to produce the antibody against the particular antigens, as well as the quantity of the antigens is directly relationship related to the amount of antibodies which is present by this living system, our own host cell or body, our living organisms. So this regulation mechanism is always necessary to regulate or control the genes. So the importance of this gene regulation is proper regulation is crucial for the normal development, cellular functions, and response to environmental changes. So based on the environmental factor, there are a lot of environmental factors are involved in this gene regulation mechanism. Okay, now let us see one level of the regulation mechanisms. So the transcription regulation are involved in this promoter as well as the enhancer is the regulatory sequencer that helps to initiate the transcription, as well as the transcription factor proteins that binds to specific DNA sequences to enhance or inhibit the transcription, as well as the epigenetic modification. Sometimes the chemical changes to the DNA or histone, like methylation or acetylation, that affects accessibility of gene for transcription. Likewise, the post-transcriptional regulation mechanisms is also called as RNA splicing, RNA stability, and microRNA. One by one, I'll tell you. RNA splicing is the removal of introns and rejoining the exons, so leading to different mRNA variants. So removal of introns is a major part in this RNA splicing. So likewise, the RNA stability, the lifespan of the mRNA can be influenced by the specific sequences and protein affecting how much proteins is produced. So the RNA stability is mandatory because there are so many factors are affecting the proteins. Likewise, the microRNA is small RNA molecules that bind to mRNA and prevent the translation. So these are the way we can able to regulate the post-transcriptional regulation transcription process. Likewise, the translational regulation. Now let us see the translational regulation. So ribosome binding, the efficiency with which ribosomes bind to mRNA can increase the protein synthesis. So ribosomes also one of the major regulation mechanisms in this translation. Likewise, the initiation factors, the proteins that helps initiate the initiate the translation and can be regulated by cellular conditions. So based on the cellular conditions, they can able to regulate by these initiation factors. Then the post-translational regulation is protein modification, also one of the regulation mechanisms. The chemical changes to the protein, for example, phosphorylation, ubiquinosing, and that can alter the activity, location, and stability. Likewise, some of the environmental factors, because the remaining things are all usually it comes under your syllabus, wherever you can see, you can get the information about the regulation mechanisms. Apart from this, some of the environmental factors are also influence this regulation mechanisms. One of the some external factors, one among the external factors is hormones, nutrients, and stress can trigger the pathway that alter the gene expression. So these are the external regulators for gene translation mechanisms. That means the protein synthesis. Likewise, the cellular context, different cell types express different set of the genes, so determined by the specific function and needs. So each and every cells are having some unique regulation mechanisms. For an example, lung cells and liver cells or kidney cells or muscle cells, whatever it is, each and every cells having some unique regulation mechanisms. So based on the cell types, also the protein synthesis will take place. Protein regulation mechanisms will take place. So by this gene expression regulations, vital process with a multiple layer to ensure the genes are expressed in the right time and the right amount. That's a very essential. So when it's required and what amount of the proteins are required, based on that, they should regulate inside our body. So now let us see the gene expression versus gene regulation. There are small minor difference between these two. Gene expression, also gene regulation. Both are similar together, but the sum of the point, they have some variations. So now let us see gene expression. This process by which the instruction on our DNA are converted into functional products, such as a protein. Is a definition of gene expression. But in the gene regulation, the number of mechanisms, the process involved in this turning genes on and off to ensure appropriate expression of genes at the proper time. So gene expression, it's a study of expressing the DNA to protein. This process is called gene expression. But the gene regulation mean already told you is the on-off control mechanisms. Whenever they require, they should express the protein, as well as whenever they need enough proteins are produced by these genes, they have to stop the process of this translation or transcription process. It's a two-step process. Gene expression is a two-step process. That means transcription as well as the translation. But in the regulation mechanisms occur either at the transcriptional process or post-transcriptional and translational process and post-translational level. So any level, the regulation mechanisms may take place. But in the gene expression, only two major steps are there. Only the gene expressions are classified into two types: only transcription and translation. But the regulation mechanisms take place in different way. There are 4 different locations they can able to take place the regulation mechanism. That means the transcriptional, post-transcriptional, and translational and post-translational level. The structural elements are introns and exons, but here the structural elements are in gene regulation is the initiation site, promoter sites, and enhancer and silencer. There are 4 structural elements are there in this gene regulation mechanisms. So the gene expression is responsible for the synthesis of gene products. But here gene regulation is responsible for controlling the amount of proteins or amount of product and the types of gene products based on the requirement of the cells. So based on the requirements, they should produce. So otherwise, they have to regulate the amount of productions which is required based on these cells. The decision is taken by the cells and its memory cells and all. Okay, but the gene expression mean it's the process of synthesis of the gene products. This is the only mechanisms takes place by this gene expression. So this is about my session today. If you have any doubt, you can able to ask. So,
Prof. Prakash, thank you for the lecture. We have one question from the student in the meeting chat.
yeah, okay. What are the key regulatory elements, the promoter regions? Okay, yeah, 1 second. There are so many key regulators already told you. One second, I just share the slides. Sorry, I didn't mention here. Okay, 1 second. Okay, actually, the core promoter elements are Tata box and initiator sequences like TSS and GC box. These are the core promoter elements which is required for this regulation mechanisms. Likewise, the transcription factor. During the transcription factors, general transcription factors like TFIA, transcription factor 11A and 11B, 11D are essential the regulatory mechanisms. Like with the specific transcription factors like NK, natural killer cells, NF, C-mycin. There are so many proteins also involved in this kind of promoter region, regulation mechanisms in the promoter regions. So the another question is genome mapping assists identifying the regulatory regions that control gene expression during the immune response species. Yes, of course. How does the genome mapping assist in identifying the regulatory regions that control the gene expression during? Yes, of course, we can. That's why we are doing the genome mapping. So genome mapping, the main purpose of doing the genome mapping is helps to control by our own being. So control the level of gene expression, or we can able to increase the or we can able to increase the synthesis of our own particular required protein level. For an example, if you want to increase the growth of the fish, so that we can growth hormones, we can able to induce the amount of protection by modification in these promoter regions or related regions. We can able to induce the process of the transcription and translation process so that they can able to produce high amount of the hormones. So they can able to produce more amount of hormones. So obviously it helps to increase the growth of fish or increase the antibiotic ranges or whatever it is. Any protein we can able to induce by this way. So next question, how can genome mapping techniques in individual or population be used? Okay. Population. Precision medicines, of course. Genome mapping techniques in individuals or populations to be used. So of course, so nowadays in future, already in the last session itself, in the bioinformatics classes, I told you the personalized medicines. Each every persons or animal or species, we can able to design the specific individual medicines for the individual persons, not based on the generic medicines we are not going to use in future. So based on the genome mapping, we can able to design our own drugs for our own disease. We can able to it's possible just because of this genome mapping to understanding the genes that actually the problems occur. For an example, I already told you in the last class string analysis. You may, if you are attend my class classes, you may heard about the string analysis. The network of genes responsible or indirectly involved in this expression studies or functional analysis. So by understanding this to functions or malfunctioning of the particular protein, that may not be takes place by this protein. It's maybe the reason for some other protein which is directly or indirectly helpful or supporting the protein, affecting or malfunctioning of that protein may affect the function of this protein. So we can easily identify by using the string analysis by genome mapping with the help of string analysis or bioinformatics tools. We can design our own precision or individual medicines for the individual cases. Case by case, we can able to design the drugs. So in future, it's all nothing now. Even nowadays, also start doing this kind of research. So now I achieve the results. So it's all easy. It's all very easy possible by using the clinical data. Already told you, but unethics also there. Some of lot of unethical things will take place just because of the whole genome mapping, genome process and that. Because everything is known by everyone. Because in case if I have our genome data, I cannot able to go for the insurance or I cannot able to go marry someone. In case if I have any genetic disorder, I cannot able to go for any loan approval or insurance. There are a lot of things that happen in future. That's why government are not providing any legal rights for this kind of research nowadays. These are major challenges by means for implementing this research in all the human being. But in animal species, we can do. That's not a problem. We can able to design by our own clinical data.
The last question, Prof. Prakash. Maybe can you tell us or give the example the most simple application of the gene expression and regulation related to water environment stress or related to fish disease, so the student can easily apply in their research, or maybe they will adapt to their thesis.
Yeah, definitely. Already I told you, to understanding this kind of basic gene expression analysis, we can able to do lot of genetic engineering process. If you want to modify our own species, if people are working in the aquatic organisms or any organisms, even it may be a plant or animal or aquatic microbes or animal species, whatever it is, the regulation mechanisms is very important to understand, to control over the species. So then only we can able to increase the growth rate, or increase the we can able to produce the medicines against any disease or disorder, or we can able to design the new species with modification, be altering the genes or inserting the genes to some other species, gene transfer mechanisms. Everything it's need for this gene expression as well as the gene regulation mechanisms. To basic understanding, without this basic understanding of the cell biology, molecular DNA and RNA structure and functions, as well as the gene transcription translations, that means the central dogma theory, then the gene expression and regulation mechanisms. These are the basic fundamentals for each and every one. So without knowing, understanding this gene expression, not only gene expression, from the cell biology, cell structure and functions, the types of the cell, nature of the cells, where it will grow, function of the cells. Likewise, the internal organelles, then the molecular level structure and function of the DNA and RNA, chromosomal, plasmid DNA, etc., etc. Then the molecular biology, the basics of DNA, restriction sites, restriction enzymes, and vectors, plant vector, animal vector, or space based on this, all this kind of molecular biology. Then the genetic engineering, transformation, or particle bombardment. Before that, we must know the molecular biology, yeah, of course, gene expression, transcription, translation, and replication, and all. Then the regulation mechanism, post-translation, transcription, modification, and the promoter of the operator regions. So then only we can able to go for the genetic engineering. Once if you understand the genetic engineering, then we can apply this genetic engineering wherever we need. Even it's maybe aquatic research or animal research or medical research or human or whatever it is. So these are the basic things for everything. For molecular level modification, genetically modified organisms, if you want to synthesis any GMO.
Thank you, Prof. Prakash. Hopefully, after this lecture, the student will have a new idea for the research, and maybe they will make some proposal related to the standard of molecular biology or basic mechanism of RNA and another protein, so they can solve the problem about the water stress or environmental stress, or maybe they will get the new medicine, or will expand the growth rate, and some research about the aquatic resource and management. Before we close this program, maybe Prof. Un will give a speech.
Thank you, Ms. Lutfi, for the guidance for the leader for the join in the 3-in-1 program. So I hope Prof. Prakash will be joined again for on the 28th September. Yes, thank you so much, Prof. Prakash. And thank you so much, Mbak Lutfi, and thank you so much for audiences in the student program in Aquatic Resource Management program. So thank you so much. I will return to time in Mbak Lutfi. Thank you.
Thank you, madam. Thank you, Professor Woon. Thank you, Lutfi. Thanks for
Yes,
giving this
thank you,
wonderful.
Prof. Prakash.
See you later on Saturday, Prof. Prakash.
Yes, sure, ma'am.
Maybe
Sure.
with me or with another lecture in aquatic resource management. Adik-adik, saya ingin ini ada apa namanya link presensi kehadiran ya, silakan diisi dan post test. Sebenarnya apa yang diceritakan oleh Prof. Prakash itu tadi adalah basic ya, basic dari bagaimana sebenarnya keadaan protein kita gitu ya. Genome expression itu bagaimana ceritanya. Nah, kalau hanya membaca dari jurnal memang akan sulit sekali memahami, sehingga seperti yang dicontohkan Prof. Prakas tadi, memang dalam tahapannya itu lebih bagus kalau teman-teman belajar melalui YouTube, sehingga bagaimana perpasangan dengan ini, translasi itu bagaimana dalam ini, kenapa begitu gitu ya, itu biasanya akan lebih mudah dipahami kalau teman-teman belajar lagi melalui YouTube, dan itu benar-benar harus diulang-ulang. Nah, karena apa? Karena ini memang basic teman-teman. Jadi kalau teman-teman bisa memahami bagaimana gene expression gitu ya, lalu fungsinya apa gitu. Nanti kalau teman-teman paham tentang ini gitu, akan lebih mudah untuk nanti misalkan ingin S2 tentang penyakit gitu ya. Nah, membuat obat untuk penyakit itu harus sesuai dengan ininya kan, apa namanya, dengan tujuannya gitu ya, tadi dibicarakan. Nah, dengan teman-teman tahu gene expression itu akan menembak secara langsung sehingga akan lebih tepat gitu ya. Lalu misalkan nanti teman-teman akan berbicara tentang menaikkan grow rate gitu ya. Menaikkan grow rate, memilih indukan yang bagus. Nah, indukan yang bagus nanti bisa dimodifikasi kalau teman-teman tahu bagaimana basic-basicnya memodifikasi indukan yang bagus. Lalu misalkan yang berpenyakit gitu ya, diobati penyakitnya ikannya, bagaimana dia lebih cepat sembuh dengan ilmu-ilmu yang sudah didapatkan tadi begitu. Dan modifikasi-modifikasi yang lain itu bisa kita gunakan tidak hanya di aquatic management, tapi misalkan di animal, di plants, atau di manusia, di cancer, semua semua apa namanya, semua elemen itu punya punya komposisi yang tadi itu gitu ya, walaupun ada beberapa gen yang tidak berekspresi. Walaupun begitu, tapi semuanya itu bisa kita atur gitu teman-teman, kalau kita bisa memahami yang apa namanya, gene expression tadi, sehingga bisa kita modifikasi sesuai dengan tujuan dari penelitian kita begitu. Sehingga sebelum memodifikasi, teman-teman harus tahu empat langkah atau empat ekspresi yang memang tadi menjadi fundamental begitu ya. Silakan apabila ingin ditanyakan.
Kalau tidak ada pertanyaan, coba dikasih tugas Mbak Lutfi. Ya, kayaknya ya mungkin kalau dari sisi basic teori tadi, mungkin adik-adik kurang paham ya untuk menerimanya gitu. Jadi memang harus ada contoh dan aplikasi. Nah, nanti ketika di sela-sela yang ada Pak Ekwan ya, ada Pak Ekwan, ada Bu Lutfi, nanti ketika menjelaskan setelah 3 in 1 selesai, nah mungkin nanti misalkan delesi gen itu kapan digunakan, kemudian modified gennya kapan digunakan, kemudian saat menempelkan gen dan sebagainya. Mungkin karena ya itulah tuntutan ya adik-adik mahasiswa, apapun yang terjadi sekarang ini ya kita harus mengikuti. Kalau kita tidak mau belajar bahasa Inggris, artinya kita tidak bisa mengikuti perkembangan ilmu dan teknologi zaman sekarang. Yang mungkin nanti sambil aplikasinya, yang penting teorinya sudah diberikan. Artinya kalau kita sudah terbiasa untuk mengaplikasikan dan mempraktekkan seperti pada saat praktikum isolasi DNA, kemudian bisa membayangkan seperti apa DNA itu seperti apa, kemudian cara memotongnya seperti apa, kemudian amplifikasinya seperti apa, itu tentunya lebih mudah. Seperti kakak-kakak kelas ya, yang kakak-kakak tingkat itu ketika belum praktikum tidak bisa membayangkan. Tapi ketika sudah praktikum, nah itu lebih paham, dan artinya banyak sekali dari adik-adik di program studi MSP yang memang ya tertarik untuk belajar lebih dalam lagi tentang biologi molekuler lingkungan. Ya itu saja, terima kasih Bu Lutfi, monggo dilanjutkan.
Harapan saya teman-teman, memang apa namanya, fakultas kita belum punya alat-alat yang menunjang untuk kegiatan praktikum eDNA gitu ya, atau PCR, GE sampai ke sana. Akan tetapi dengan keterbatasan itu jangan lalu menjadikan kita tidak belajar, menjadikan kita itu tidak tahu perkembangan zaman. Bahkan yang diceritakan Prof. Prakas tadi itu adalah sangat basic sekali yang seharusnya untuk level sekarang yang sudah ada ChatGPT, AI gitu ya, itu sudah harus kita ketahui bersama gitu. Bahkan sekarang ada ChatGPT yang tanpa kita apa namanya datang ke laboratorium itu sudah ada tools ya yang kita bisa apa namanya belajar gitu ya langsung hands on virtual itu sudah ada yang seperti itu Prof Uun sehingga eh, apa kita seolah-olah itu sudah ada di laboratorium terpadu yang risetnya sangat molekuler
Live.
gitu ya itu
Yeah.
ya itu sudah ada sekali sehingga saran saya untuk teman-teman mahasiswa begitu ya kalau belajar itu jangan hanya yang dibuka TikTok, Instagram dan lain sebagainya tapi bagaimana perkembangan riset yang terkini terkait tentang perikanan misalkan apa news-news update yang terbaru atau apa namanya perkembangan yang sedang hits, advance teknologi yang sedang hits itu yang menjadi concern kita bersama sehingga tidak hanya di kelas saja kita belajar tapi via handphone atau via teknologi yang sekarang itu kita sudah bisa apa namanya belajar dengan profesor di luar negeri, belajar dengan teknologi yang sebegitu canggih. Nah, itu bisa dipelajari dengan gratis kan bahkan hanya dengan kuota. Nah, itu saya harap bisa di apa namanya dimanfaatkan oleh kita bersama. Jangan jangan hanya karena di lab hanya mengukurnya kualitas air saja lalu apa mahasiswa MSP hanya able to mengukur kualitas air ya jangan banyak segitu gitu ya. Molekuler juga harus tahu gitu. Protein, karbohidrat di biokimia juga harus dipelajari. Nah, itu apa namanya saran saya untuk adik-adik mahasiswa seperti kegiatan ini gitu ya terasa sangat tidak ada yang menarik tidak tertarik padahal program ini itu memang benar-benar dibuat oleh fakultas atau universitas itu agar teman-teman dengan duduk saja di kelas itu bisa seperti kuliah di luar negeri kan begitu dengan profesor-profesor yang mumpuni di bidangnya, profesor yang memang terkenal di bidang bioteknologi ada siapa, nanti di bidang lingkungan ada siapa. Nah, kita difasilitasi dengan ini gitu sehingga saya harap ini dimanfaatkan sebaik mungkin. Kalau ada pertanyaan, gitu ya, silakan bertanya, jangan hanya eh, join lalu tidak mendengarkan. Nah, itu akan sayang sekali. Saya rasa untuk teman-teman.
Tidak ada yang bertanya ya Mbak Lutfi ya. Ya, besok lagi nih harus di absen satu-satu nih Mbak Lutfi.
Iya, apalagi hari Sabtu, Prof. Uun.
Ya, harus di absen satu-satu nih.
Yang
Tidak ada
misalkan
pertanyaan.
terkendala bahasa, misalkan boleh kok apa namanya bertanya dalam bahasa Indonesia di chat box gitu ya, nanti di translate itu sangat boleh sekali teman-teman.
Apalagi nih ya, handphonenya adik-adik kan canggih tuh ya. Langsung ada trans Google yang pakai apa itu pakai layar tuh loh ya. Saya kan oh ini canggih-canggih nih adik-adik mahasiswa baru nih. Handphonenya bisa langsung trans Google ini loh. Jadi kalau misalkan suaranya apa langsung dimasukkan atau langsung ini tadi slide-nya itu langsung difoto, langsung masuk di Google Translate. Ya. Nggak ada pertanyaan, Mbak Lutfi? Ya, kalau saya senang sekali ya tadi begitu tahu basic respon. Jadi ketika Lux Opron itu membuka, berarti misalkan kenapa terjadi apa namanya? Misalkan ekspresi gennya itu ada di sequence yang dimulai dari timin, kemudian nanti sebelahnya adenin, sebelahnya guanin. Kenapa bisa seperti itu? Nah, ini saya rasa adik-adik ya, kalau adik-adik betul-betul belajar, betul-betul eh, menjiwai. Jadi kalau tidak tahu itu enggak apa-apa, cuman harus membaca. Jadi kuncinya itu adalah membaca. Jadi tolong nanti hasil rekamannya didengarkan lagi 1 kali, 2 kali, 3 kali, 4 kali seperti itu. Ya saya pun walaupun artinya sudah mengajar berkali-kali, kalau tidak belajar ya tidak bisa cerita gitu. Artinya ketika sekarang misalkan adik-adik belajar tentang bioassessment, misalkan ya saya kasih contoh, misalkan Dokter Umi sekarang sedang menguji merkuri ya, saya contohkan aja merkuri, kemudian diujikan pada artemia. Nah, pada 48 jam ketiga, jadi 24 jam kali 2, 48 jam, ternyata artemianya itu mati. Nah, sekarang yang menyebabkan mati itu apa? Mesti adik-adik menjawab, contohnya adalah merkuri. Tapi matinya itu kenapa? Kan itu ya, jadi ketika merkuri itu menjadi prekursor, stresor di artemia, di sel artemia, nah itu yang menyebabkan matinya itu apa? Jadi merkuri itu punya binding site. Jadi di unsur atau di senyawa merkuri yang berikatan dengan H2O di dalam buffer itu ya, yaitu nanti ketika diujikan di artemia nanti akan mengikat bagian site atau asam amino atau protein di bagian prekursornya artemia. Kalau asam amino yang ada di artemia itu tidak diikat oleh bagian binding site bondnya si merkuri, nah ini artemianya bisa nggak mati, Dek. Nah ini contohnya. Tadi kalau misalkan Prof. Prakas bicara tentang ekspresi gen, kapan toh ekspresi gen itu terjadi? Nah ini contoh yang paling sederhananya yang nanti kita akan lakukan untuk pengabdian. Nah itu adalah menguji merkuri atau herbisida ya terhadap artemia. Jadi dilihat kenapa 24 jam, kenapa 48 jam, kenapa 78 jam, sampai 96 jam. Jadi kalau 96 jam mati semuanya, nah itu apa yang mengakibatkan merkuri itu toksik? Karena ada ikatan kovalen di bindingnya si merkuri yang berikatan dengan H2O di side sisinya itu berikatan dengan asam amino X di artemia. Sekarang kita belajar ekspresi gen di lingkungan. Nah, artemia itu punya protein apa? Contohnya, misalkan dia punya timin yang sensitif terhadap prekursornya Hg atau merkuri, nah ini dia akan menjadi lemah sehingga transkripsi gen di artemia itu tidak bisa berjalan sehingga selnya menjadi lemes dan mati. Nah, itu contohnya ya. Jadi, ya itulah kalau kita tidak tahu ilmu dasarnya, ya kita akan tertatih-tatih. Tapi tidak usah khawatir karena banyak sekali yang harus kita bisa baca ya dengan teknologi sekarang itu gampang sekali. Ya, saya rasa itu contoh yang bisa adik-adik renungkan apa sih yang bisa saya dapat hari ini ketika saya belajar tentang ekspresi gen, tentang asam amino, tentang nah kalau kita sudah tahu banyak, nah nanti kita akan tertarik dan kita akan bisa mengerjakan sesuatu dengan banyak hal. Contoh lagi apa herbisida ya misalkan herbisida itu kalau misalkan terbuang ke sungai, nah itu kan hanya 3 ya, berbau, berasa atau berwarna. Tapi bagaimana efeknya terhadap ikan, terhadap bentos, terhadap plankton? Nah itu nanti masuknya lebih dalam lagi ya ke molekuler, bisa ke proteinnya, bisa ke DNA-nya. Belum lagi tentang environment DNA. Jadi kita tidak usah mengambil sampel yang gede-gede itu, tapi kita dengan mengambil sampel air saja, nah itu kita sudah bisa mendeteksi ada DNA apa saja yang ada di situ. Apakah DNA-nya ikan yang lewat di situ, bentos, plankton, bakteria, dan sebagainya. Sekarang yang lebih berkembang lagi, terbaru yang kita harus pelajari itu adalah mikrobioma. Jadi kita mengambil sampel air misalkan 1 mililiter ya, tapi kita bisa mendeteksi bisa 20, bisa 30 DNA daripada spesimen. Ya nanti sambil berjalan nanti kita kenalkan ya Mbak Lutfi. Ya biar adik-adik punya gambaran dan punya semangat untuk bisa lagi belajar lebih banyak di program studi MSP. Saya rasa itu Mbak Lutfi.
Sebenarnya mahasiswa ini semangat-semangat saja, Prof. Uun, tapi pengetahuannya teman-teman ini terbatas. Misalkan biomarker, kan taunya biomarker itu misalkan hanya bentos, kan begitu ya teman-teman ya.
Betul.
Nah, bentos itu kalau teman-teman taunya hanya bentos, itu ibaratkan sekarang sudah zamannya iPhone 16. Teman-teman tuh masih Nokia 3610 itu loh yang ringing tidak cuman bisa SMS itu sampai segitu.
Iya.
Nah, teman-teman harus tahu bagaimana teknologi-teknologi terbaru. Misalkan biomarker sekarang seperti yang dikatakan Prof. Uun tadi tidak perlu cari bentosnya, tidak perlu ukur kualitas air segitu banyaknya. Kalau teman-teman tahu transkriptomik misalkan, itu memungkinkan identifikasi perubahan ekspresi gen yang bisa mendahului indikator-indikator yang tradisional, indikator toksisitas tradisional misalkan gitu ya, mengukur segala macam bentosnya, planktonnya. Nah, itu bisa disederhanakan dengan transkriptomik itu tadi. Jadi memungkinkan deteksi dini dari efek kesehatan yang merugikan akibat paparan misalkan xenobiotik gitu ya. Sehingga kemampuan memprediksi kita itu lebih cepat daripada yang tradisional atau yang jadul sekali gitu ya, sehingga kita bisa mengembangkan biomarker non invasif untuk deteksi penyakit secara dini teman-teman begitu. Jadi kalau teman-teman hanya mengikuti apa namanya plankton gitu ya, kualitas air atau bentos dan lain sebagainya, itu ya sudah jadul sekali, sudah ketinggalan zaman. Yang lainnya sudah pakai smartphone terbaru yang bisa dilipat, digulung dan di sebagainya, teman-teman masih telolet-telolet. Nah, itu seperti itu contohnya teknologi. Nah, kenapa harus mengikuti? Karena kalau tidak mengikuti setiap detik itu tuh ada apa namanya studi terbaru, sehingga kalau memang hanya terbatas ilmunya dari situ-situ saja, teman-teman tidak akan menjadi mahasiswa yang benar-benar mahasiswa gitu loh maksud saya itu. Jadi ya hanya duduk di kelas, datang, praktikum, selesai, materinya hanya sebatas apa yang disampaikan dosen gitu. Sehingga teman-teman harus mencari batas-batas ilmu yang sekarang yang advance itu sampai sejauh mana, bagaimana sel itu bisa dijadikan biomarker gitu ya, yang non invasif untuk mendeteksi penyakit lingkungan misalkan atau penyakit yang hubungannya dengan hewan, penyakit yang hubungannya dengan tumbuhan atau hubungannya dengan manusia yang lebih kompleks. Nah, itu menjadi penelitian-penelitian terbaru yang memang harus teman-teman ketahui sehingga nanti kalau teman-teman ingin menyusun proposal PKL atau skripsi gitu ya, jangan hanya judulnya seputar itu saja ya Prof. Un ya biasanya ya mahasiswa. Jadi yang lebih advance gitu ya, sesuai dengan perkembangan zaman yang ada gitu. Jadi jangan yang di-upgrade dari teman-teman itu jangan hanya handphonenya saja, jangan hanya style berpakaiannya saja, jangan hanya mungkin tren make up-nya, akan tetapi isi dari otaknya itu juga harus ikut berkembang. Apa yang sedang hits, apa yang sedang tren itu diikuti, bukan hanya hot gossip gitu ya, tapi hot news yang berhubungan dengan studi program, misalkan hot news yang berhubungan dengan peminatan teman-teman. MSP ini kan sebenarnya luas sekali, teman-teman. Misalkan minatnya ada di bidang ilmu A gitu ya. Nah, itu yang akan dipelajari begitu. Sehingga nanti saat pembagian apa namanya dosen pembimbing skripsi, teman-teman sudah siap. Oh, saya minatnya seperti ini. Yang penelitian terdahulu seperti ini, sehingga penelitian sekarang yang terkini gitu ya, saya mengusulkan seperti ini. Itu akan menjadi apa namanya lebih baik untuk teman-teman.
Ya, nilai lebih ya artinya. Ya, contohnya kemarin ini Mbak Lutfi ada yang tes di HRD Cimory di Jawa Barat ya. Jadi kemarin itu saya membimbing saudara Raihan Surya angkatan 2020. Nah, ketika sudah wawancara itu ternyata kami ditelepon dari Jawa Barat Nah, itu dari Direktur HRD itu menyatakan benar apakah tidak bahwa mahasiswa ibu melakukan ini dan sebagainya. Nah, itu saya sampai kan itu kemarin kami di kabar itu sekitar setengah 12 malam ya. Artinya kalau di sini mungkin pagi ya. Nah, itu ternyata dikonfirm betul nggak bahwa ini melakukan ini kan adik-adik kemarin melakukan analisis nanopartikel ya di sungai. Dari cemaran sungai itu ada yang bagian nanopartikel, ada yang bagian respon stres individunya, ada ikan, ada udang, ada zooplankton dan sebagainya. Ternyata itu dikonfirmasi sampai sebegitunya saya juga kaget ini saya dapat nomor telepon dari mana kan saya juga nggak tahu gitu kan. Ternyata nah di situ si Raihan itu menuliskan bahwa tugas akhirnya adalah ini melakukan ini dan sebagainya. Nah, itu ternyata saya pembimbingnya dikonfirmasi. Nah, ini yang bisa saya sampaikan ke adik-adik ya, misalkan adik-adik sekarang menganalisis kualitas air. Nah, itu adalah basically memang harus dianalisis kualitas air itu. Tetapi di dalam kualitas air itu ada apa? Misalkan adik-adik belajar tentang biomonitoring. Nah, mediumnya apa? Air benar. Tetapi biomon apa yang dimonitoring di situ. Nah, sekarang kalau misalkan memonitor sebuah biota, apakah itu bakterianya, apakah itu zooplanktonnya, ataukah fitoplanktonnya terkait dengan pencemaran misalkan. Nah, kenapa si zooplankton itu ada di situ ketika air ini tercemar? Jadi bukan hanya menganalisis kualitas airnya saja. Jadi tinjaulah dari sisi pencemarannya itu seperti apa. Misalkan kalau adik-adik meneliti tentang plankton, masuk lagi ke biologi molekuler lingkungan. Kenapa di situ terjadi blooming fitoplankton? Biomolekulernya ada di mana? Misalkan pembelahannya planktonnya terlalu cepat. Kemudian apa yang menyebabkan terlalu cepat itu? Misalkan ada nitrit sama fosfat. Nah, sekarang nitrit itu loh membuat apa toh kok bisa menjadi pemicu atau prekursor daripada plankton itu menjadi lebih cepat 3 kali sampai 4 kali lebih cepat pertumbuhannya. Nah, seperti itu deh ya. Jadi contoh-contohnya yang bisa dikembangkan dan ide-ide kreatif itu seperti apa. Adik-adik bisa juga mengeksplor dari handphonenya masing-masing, dari smartphone-nya ya. Kalau adik-adik tidak bisa kreatif ya, eman deh kamu nggak dapat apa-apa nanti. Ya, dapatnya ya hanya dikasih dosen aja, tapi adik-adik tidak bisa mengembangkan. Jadi kalau medium air ya memang mediumnya air iya, tetapi apa yang adik-adik bisa lakukan di situ? Misalkan kalau pencemaran, kenapa Hg itu bisa mencemari perairan? Apa yang dicemari? Nah, itu yang dibahas sisi detailnya seperti itu. Gitu nggak Mbak Lutfi? Ya,
Terima kasih Prof. Uun, insight barunya. Saya rasa hari ini bisa cukup sekian. Saya berharap nanti adik-adik mahasiswa bisa lebih excited untuk mengikuti gitu ya. Karena kalau teman-teman kuliah di tempat lain belum tentu mendapatkan guest lecture dengan mendatangkan profesor. Ini zaman saya dulu teman-teman ya, sebelum COVID itu susah sekali. Sehingga COVID itu banyak hikmahnya sebenarnya. Dengan COVID kita punya banyak tools-tools yang bisa kita gunakan gitu ya. Belajar bisa daring, luring, lalu kuliah bisa kuliah dari mana saja. Banyak sekali pokoknya yang bisa kita dapatkan. Salah satunya adalah mendatangkan profesor-profesor yang ahli di bidangnya, yang kita hanya duduk bisa mendengarkan gitu ya. Ini sangat-sangat kesempatan yang sangat bagus sekali. Dulu mungkin kita mendatangkan profesor dari Belanda itu perlu ratusan juta ya Bu Un, misalkan
iya,
gitu ya.
betul.
Sehingga 1 tahun itu mungkin hanya 1 kali. Nah, sekarang
Iya.
1 tahun itu bahkan teman-teman kalau lihat pamflet di FIK itu bisa sampai 30 sampai 40 profesor dari luar negeri yang datang ke kita hanya untuk memberikan guest lecture atau kegiatan yang lainnya. Itu saya rasa harus benar-benar dimanfaatkan oleh teman-teman sekalian gitu. Kita bertemu lagi hari Sabtu, teman-teman. Saya akhiri kegiatan program 3-in-1 kita hari ini. Selamat siang. Wassalamualaikum warahmatullahi wabarakatuh.
Iya, waalaikumsalam. Terima kasih banyak Bu Lutfi. Iya, terima kasih.